Hello everyone, in this video we will discuss about H. pylori gastritis. Now what is H. pylori? H. pylori is a bacteria and is a gram-negative bacteria. And in this picture we can see this is a H. pylori bacteria. It's a rod-shaped bacteria and has flagella which is responsible for its motility. Now the humans, they are the only known host, and also the route of spread is mostly fecal-oral. Now going to the gastritis, H. pylori infection is the most common cause of chronic gastritis. There are other causes also like autoimmune gastritis, and in the case of H. pylori, the disease has a characteristic that it predominantly affects antrum of the stomach, that it causes antral gastritis. It is associated with high acid production, and the gastrin levels, they are low, that is there is hypogastrinemia associated with it. Now going to each part. Now here we can see this is the stomach, and we can see this is the part is pyloric antrum, which is involved in the case of H. pylori. But in the later cases and in some patients, also the cardia, fundus and body can be involved. Now going to the virulence, going to the pathogenesis of the H. pylori. Now H. pylori virulence is caused by four factors mostly, like the flagella, the urease enzyme, the adhesions and the toxins which are secreted by the bacteria. Now if you remember, if this is the gastric epithelial cell, second is the layer of the mucus, and then is the layer of the acid. Okay, now the urease enzyme which is secreted by the bacteria, it converts the urea into ammonia and carbon dioxide. Now this ammonia is highly alkaline and is responsible for balancing the acidity of the stomach, and the flagella, it allows the bacteria to be motile in the viscous mucus. Now the bacteria can enter into the mucus, and the adhesion proteins, the adhesions, they enhance the adherence of the bacteria to the epithelia, and further the inflammation and the damage is caused by the toxins which are secreted by the bacteria like Kage and Vaccae. These are the toxins secreted. This is how the virulence of H. pylori takes place. Now further what happens is there is chronic antral H. pylori gastritis. Now firstly it is antrum is involved, and further this will progress as pangastritis, and pangastritis afterward will be associated with apoptosis of various cells of the stomach, leading to atrophic gastritis. It is a very important cause of chronic atrophic gastritis also. Now going to the diagnosis. Now for the diagnosis, there are three tests which are done mostly. First is the urea breath test. In this test, the patient is told to swallow urea which is labeled with radioactive carbon. Now after 10 to 30 minutes, we will detect the presence of isotope labeled carbon dioxide in the exhaled breath, which will indicate that the urea was split because the urease enzyme is present by the presence of the H. pylori bacteria. Second is the antigen detection in the stool, which can be done using PCR also. And last is the gastric biopsy. We will discuss the gastric biopsy. Now going to the biopsy. Now we know that the bacteria, they are present in the antrum. So the biopsy is preferred from the antrum, that is antral biopsy is done mostly. And when we will view this endoscopically, the mucosa will be inflamed, that is it will be erythematous, it is red in color and will have a coarse or even a nodular appearance can be seen. Going to the microscopy. In the microscopy, firstly we'll discuss about the organism. Now the organism, it is mostly concentrated in the mucus layer which overlies the epithelial cells of the stomach, and the distribution of this organism can be irregular. That means there can be areas where there is heavy colonization, and then there can be areas where there are very only few organisms. There is distribution is irregular. And lastly, we can see that H. pylori only shows tropism for the gastric epithelia. That means it is not associated with intestinal metaplasia or when there is duodenal epithelia involved. It is only associated with gastric epithelia. Going to the microscopy further, organisms. The organisms can be demonstrated on on routine stains like this one, and also on special stain like Warden starry silver stain. Here we can see this is this stain, and multiple rod-shaped bacteria, they can be seen. Now, associated with it is always an inflammation. Now, inflammation is of various types. Firstly, the inflammation, it includes variable number of neutrophils, and these neutrophils, they can enter into the epithelial cells, that is known as intraepithelial neutrophils, and even they can accumulate in the gastric pits where they can form pit abscesses. This was neutrophils. Not only neutrophils, the associated with it are large number of plasma cells and lymphocytes. Now, this is a very characteristic part of the H. pylori gastritis. We can see in this picture uh, intraepithelial neutrophils, the single arrow, this one, the intraepithelial neutrophils, along with presence of subepithelial plasma cells, these ones with the double arrow. This is a very characteristic sign of H. pylori gastritis. Okay, now we already understood that there can be increased number of lymphocytes which can be present, and these lymphocytes can increase so much that they can form lymphoid aggregates, and these lymphoid aggregates can form a induced form of mucosa associated lymphoid tissue, that is MALT, and this is responsible for lymphoma further. So H. pylori is responsible not only for gastritis, it is also responsible for adenocarcinoma of the stomach and also for lymphoma, and lymphoma also maltomas. And the H. pylori gastritis further can lead to atrophy of the mucosa. That means it can also lead to chronic atrophic gastritis later in the life. Now, these are the two main causes of atrophic gastritis. One is the H. pylori associated and second is the autoimmune. We will discuss both when I will discuss autoimmune gastritis also. Just going through, antrum is the location for H. pylori, whereas body is most commonly involved in autoimmune gastritis. Inflammation associated is neutrophils, plasma cells, where there are lymphocytes and macrophages most commonly present. And the acid production is increased, there it is decreased. Gastrin is normal to decreased over here, it is increased in the autoimmune. But we will understand each one, the difference detail, when I will discuss autoimmune gastritis also. Okay. uh, and lastly, the treatment part. In the treatment, there are we give combination of antibiotics and proton pump inhibitors, and mostly the patients, they usually improve after the treatment, although the relapses can occur after incomplete eradication or reinfection. Therefore, relapses, they are very common in these patients. This was all about H. pylori associated gastritis. The next video which I will upload is autoimmune gastritis, and then we'll discuss the difference between it also. Please like, share and subscribe to this channel. Thanks for watching this video.